It gives immense pleasure to welcome and introduction our keynote speaker of today's session, Dr. Madhusudana Rao. Dr. Madhusudana Rao, an assistant professor in the Department of Nanomedical and Polymeric Materials, School of Chemical Engineering, Yeungnam University, South Korea. He completed Doctor of Philosophy in the Chemistry, Sri Krishnadevara University, Anantapur. Thesis title is Development of Novel Stimuli Responsive Multi-Structured Polymeric Hydrogels for Biomedical Applications. Sir has 12 years of research and teaching experience as postdoctoral fellow and as assistant professor. He is a renowned expert with a proven track record of publishing groundbreaking research of 18 publications and 7 book chapters. A visionary administrator who attended 8 conferences, received 2 patents, giant researcher on 2 projects, and H-index for 41. Sir is accountable for several professional responsibilities in his career, who has handled various duties like designing, development, bioprinting, formulations of nanoparticles, synthesized drugs such as curcumin, GN25 and GH14. He is also participated in a drug delivery and gene delivery works in a collaboration with Professor Yeun from the University of Argonne, USA, and with Professor Yeungmi from South Korea. He is also received a grants, the principal investigator for the project titled Development Impressed 3D Hybrid Hydrogels for ultrafiltration bone like a appetite formation and growth factor pre-fast maturation of bone cells. Sir is playing crucial role as a giant researcher for projects titled and development of cell culture technology. Sir, we have very fortunate, we have as our guest and keynote speaker, a warm welcome to you, sir, and I request you take over the section.
Okay.
Sir Madhu sir, you will share the PPT.
Okay, I'm sharing now. Okay. So is it okay?
Yeah.
No?
Full mode.
Okay, I will go.
PPT, share your PPTs in.
Okay, now.
Yeah, now okay.
Okay. Okay, thank you for the introduction. First of all, I would like to express my heartfelt thanks to organizing committee of the FDP on recent advances in novel drug delivery applications. So my sincere thanks to my senior Dr. Swami for the kind invitation. Today I will be discussing advances in scaffold fabrication techniques using biocompatible polymers and their biomedical applications. So I have slightly modified my presentation slides to focus on more on the fundamental knowledge of tissue engineering through biomaterials. I hope those who want to start this field, it would be helpful to know basic things. So let's dive into the presentation. So here I mentioned the contents of the presentation, which includes introduction to tissue engineering, and the scaffold development for various tissue engineering applications. So let's start with something most of us have experienced, slime. Ever touched a frog or a slug? It's slimy, right? Well, that sliminess is a natural production found in our various parts of our body, like our gut and throat. The components of the slime are macromolecules like proteoglycans, polysaccharides, and collagen fibers, and they form what we call the extracellular matrix, which is found between our cells. If cells were made by air, they would be easily damaged, right? The ECM is a vital because it keeps our cells together and mobile as a group, helps to protect them from outside forces and chemicals. So ECM, it can play an important role in regulating biological functions like cell adhesion, proliferation, migration, and differentiation. So can we create an artificial ECM using biomaterials? Yes, it is possible to create an artificial ECM. In order to help, in order to help to restore or regeneration of functional tissue, even we can create a whole organ of our body. So what is the motivation behind the tissue engineering concept? Every day, tissues are lost and damaged, whether due to injury, disease, or aging. Our body constantly struggles to heal, but what if we could change that? Our dream is simple, right? To restore damaged tissues so that life goes on for our loved ones, as if a tragedy never happened. Imagine a world where no injury is permanent, where lost tissue regenerate, and where hope replaces despair. This vision is as old as human imagination itself. From ancient myths of miraculous healing to today's scientific breakthroughs, we have always sought ways to mend what is broken. But what is the challenge? The challenge is turning this dream into reality, creating functional tissue, that okay, through migration and adhesion, cells come together to form tissues, shaping the body architecture, but their role does not stop there. Cells secrete growth factors, guiding further differentiation, ensuring the precise formation of organs and systems. So it's incredible to think that from just one cell, an entire life unfolds. A heart that beats, a brain that thinks, a body that moves, all originating from a single unit of life. So it truly a wonderful life, a testament to the beauty and the power of biology. So the concept of tissue engineering is not new. If we look back at history and mythology, we find countless references to chimeras, beings formed by combining different body parts to create something new. In Hindu mythology, Lord Ganesha, he was properly grafted with elephant head, symbolizing restoration and transformation. So in Islamic tradition also, Allah Baraka was a mystical creature with the body of a horse and the wings of an eagle, representing divine synthesis. These other chimeras also, like Christian and Greek also. These stories spanning cultures and centuries reflect humanity's deep-rooted fascination with regeneration and reconstruction. Today, tissue engineering is tuning myth into reality by integrating cells, biomaterials, and technology. We are moving closer to repairing, regenerating, and even creating functional tissues. So one of the most iconic moments in tissue engineering history in the vacante mouse in 1997, a team led by Dr. Charles Vacante implanted a biodegradable scaffold seeded with cartilage cells onto the back of lab mouse. Over time, the cells grew and into the shape of human-like ear. This experiment was a breakthrough, proving that living tissues could be engineered using scaffolds from polyglycolic acid and polylactic acid, polylactic acid polymers, and cellular growth techniques. Though often misunderstood as a generally modified mouse, in reality, it demonstrated the power of combining biomaterials and cellular biology to create a functional tissue. The vacante mouse sparked both scientific excitement and ethical debates, but most importantly, it paved the way of modern tissue engineering, bringing us closer to regenerating organs, repairing injuries, and even growing tissues tailored for transplantation. From an experimental mouse to the future of medicine, tissue engineering continues to push boundaries, transforming ideas into reality. So if you see other images, even you can see here nose on forehead and ear on forearm. Scientists they have created these nose on forehead and ear on forearm in recent study. So the foundation of tissue engineering was formally defined by Robert Langer and Joseph Vacanti, who described it as tissue engineering. It is a multidisciplinary by necessity, and it is an interdisciplinary field that applies the principles of engineering, life sciences towards the development of biological substitutes that restore, maintain, or improve tissue function or a whole organ. So this definition highlights that tissue engineering is not just one discipline. So it is a fusion of multiple fields working together. Suppose medical doctors, it requires medical doctors, engineers, biologists, chemists. So medical doctors understand the clinical need and patient applications. Engineers design scaffolds, bioreactors, and mechanical systems for tissue growth. Biologists study the cell behavior, differentiation, and tissue formation. Chemists always develop biomaterials, hydrogels, and drug delivery systems. So bringing together these diverse areas of expertise allows us to create functional tissues and organs, pushing the boundaries of regenerative medicine and transforming healthcare as we know it. So why tissue engineering important? So as we know, organ transplantation has been a life-saving solution for patients suffering from organ failure, like a heart, kidney, whatever it is. So vital organs like heart, kidneys, liver, lungs, pancreas, intestine, and thymus can be transplanted to replace damaged or non-functional organs. However, despite its success, organ transplantation faces two major challenges, like a severe shortage of donor organ, severe shortage of donor organ. So every year, the number of patients on the waiting list increasing, but donor availability remains limited. Donor availability remain limited. The need for lifelong immunosuppressants. Transplant recipients must take these some drugs like to prevent rejection, increasing the risk of infections and other complications. So that's why tissue engineering plays a crucial role by developing lab-grown tissues organs. We aim to overcome donor shortage, reduce the dependency on immunosuppressants, and provide personalized solutions for patients in need. Tissue engineering is not just a scientific advancement, it is a necessity to address the growing organ failure crisis and revolutionize regenerative medicine. So if you see here the basic principles of tissue engineering. In tissue engineering, we require the three important components: cells, scaffolds, and signaling molecules. So as we know, cells we can collect from our own body, and scaffolds we can make from polymers, like either natural polymers or synthetic polymers. Signaling molecules also can play an important role to promote the cell growth and adhesion and even differentiation. So signaling molecules are mostly growth factors. These are our growth factors. So if we combine these three components, so we can able to regenerate tissue, even whole organ, with appropriate environment with respect to time. So if you see here this image, so in tissue engineering, first we need to isolate cell from the patient, and then we have to expand the cells under in vitro conditions. So meanwhile, we have to use a scaffold with a three-dimensional architecture. So when we seed these cells, isolated cells on scaffold, under particular time and appropriate environment, the cells grow within this scaffold, like 3D scaffold. So once these cells grow or maturation before implantation or after implantation, so we can restore or regenerate the functional tissue. So this is the basic principles of tissue engineering. So we need to use three major important components: cells, scaffolds, signaling molecules. So if you come to the cells, what type of cells are important? That is very, very important. So, mostly we use stem cells. So why we use stem cells? So as we go through our lives, each of us will have very different needs for our own healthcare, right? Scientists and doctors are constantly researching ways to make medicine more personalized. One way they are doing is by researching stem cells. Stem cells are cells that are undifferentiated. Stem cells are cells that are undifferentiated. Um, Meaning they do not have a specific job or function, while specialized cells like skin cells, muscle cells, nerve cells, they do have specific job. Suppose skin cells, they protect our body. Muscle cells, they can contract. Nerve cells, they can send signals. However, stem cells, they do not have a specific structure of function. Stem cells, they do have a potential to differentiate into specialized cells. All type of specialized cells like skin cells, blood cell, fat cell, liver cell, nerve cell, bone cell. So if we use stem cells, we can able to produce specialized cells. It means, as we know, tissue, it is a collection of cells and extracellular matrix. So specialized cells and extracellular matrix. If the cells die, so where new cells come from? The stem cells can play an important role to produce the specialized cells. So stem cells, they can self-renew throughout our life, and also they can differentiate to replace the damaged or diseased specialized cells. That's why cells are very, very important. Stem cells are very, very important for tissue engineering. So what is the cell difference? So we have to know about cell differentiation, because stem cells, they can differentiate into various type of specialized cells, all type of specialized cells. So what is the cell differentiation? As cell goes through the multiple cell division, it is going through various developmental stages like zygote or embryo. So each development stage, cells begin to differentiate and become specialized to form a tissue, right? So the meaning of differentiation, it is a process of cell becoming specialized in structure and function. So once it become like specialized, it produce specialized cell, the cells has its own function, like a skin cell, they protect our body, and a muscle cell, they can contract, and nerve cell, they can send signal. They have a specific job. So why self-renew and differentiation of stem cells? So in our body produces, in general, in our body produces new billions of new blood cells every day, right? Blood cells have a limited, because blood cells have a limited lifespan. So the body needs to replace them quickly and often. So stem cells are building blocks of blood cells. Stem cells in the bone marrow can become any type of blood cells in order to replace the damaged cells. So all human, a supply of adult stem cells that constantly repair or replenish the rest of the cells in the body. So without stem cells, very difficult to survive our life, very difficult to go through our life. Throughout our life, we have a pool of stem cells always. So that's why stem cell always self-renew in order to maintain the stem cell pool and also differentiate into more specialized cells. So in order to replace the dead or damaged cells throughout our life. So if you take some example like so if you take an example like small intestine. So in intestine, the cells are continuously die every day. So in order to replace these damaged cells, the stem cells always differentiate to produce like specialized healthy cells. healthy cells to maintain the tissue function. So that's why using stem cells to replace damaged body tissue is called regenerative medicine. If we supposed to add these cells on 3D scaffold, it is structurally similar to native tissues, thereby regenerate functional tissue. So as we know, stem cells we can found in embryonic stem cell. We can found in early blastocyst. It is a inner early blastocyst. So in a blastocyst, we can found embryonic stem cells. So embryonic stem cells are pluripotent cells. They can differentiate into all possible types of specialized cells in our body. So in order to produce like a specific kind of specialized cell, it requires a particular condition. Suppose if we apply a condition A, it produces skin specialized cell. If we apply condition B, it produces like neurons. If we apply condition C, like produce blood cells. If we apply condition D, it produces liver cells. All these are all specialized cells. So each in order to produce each specialized cell, it requires a specific condition. More specific cell, it requires specific condition. So it is a more challenge in tissue engineering. So in the tissue stem cells also we can found in our body throughout our life, like on the surface of eye, brain, skin, breast and intestines, testicles, bone marrow and other muscles. So usually blood stem cells we can found in bone marrow. On differentiation produce like specialized types of blood cell like red blood cells, white blood cells and platelets. So other important term, induced pluripotent stem cells, IPS cells. So in general, the use of embryonic stem cells are not ethical because these cells collect during the early pregnancy time, because these embryonic stem cells found in a blastocyst. So it is not ethical. That's why the scientists always focusing on changing like procedures to prepare like in laboratory, the stem cells prepared in the laboratory conditions. So the stem cells are known as induced pluripotent stem cells. These stem cells behaves like embryonic stem cells. So in the laboratory, these induced pluripotent stem cells on differentiation produce all possible types of specialized cells. The main advantage is there is no need to use of embryos. It is not ethical. So how we can generate these IPSCs, induced pluripotent stem cells? So if we take cell from the body, like a more specialized cell, and then induce genetic reprogramming, and induce genetic reprogramming, we can able to get induced pluripotent stem cells. So these cells behaves like embryonic stem cells. It can able to produce all possible types of specialized cells. So you see here a single cell, a cell collecting from the body, skin cell, and applying genetic reprogramming. Then we can able to produce like pluripotent stem cells, IPSCs. On differentiation, you can see various type of specialized cell like cartilage, bone, and neural tissue, muscle, and other like respiratory epithelium. So we can able to produce all type of tissues. So there is no, in this case, there is no use of there is no need of embryonic stem cells. So as we know, cells are more important for tissue engineering. So other component is scaffolds. Scaffolds also important for tissue engineering. So in general, scaffolds, these are like a three-dimensional porous structure. In order to provide tissue, in order to provide developing a tissue and allow cells to adhere, proliferate, differentiate, and importantly to secrete ECM to produce functional tissue. So it should be like a three-dimensional porous structure. It should act as a template for tissue growth. Because in our body, we have a lot of tissues, each tissue having its own extracellular matrix. So if we create artificial three-dimensional polymeric scaffold, so that structurally it is similar to the native extracellular matrix. So that's why we need like a three-dimensional porous network structure. It could allow the cell adhere, proliferate, differentiate, and importantly secrete ECM to produce functional tissue. So when we seed cells on this scaffold, so it should be biocompatible. As everyone know about biocompatibility, it should not toxic to the our native tissues. So the scaffold should have an interconnected macroporous network for vascularization, tissue ingrowth, and nutrient delivery. Because once we seed cells on the particular scaffold, so the scaffold should grow and attach, and even differentiation. In order to perform this function, so it requires like a porous network structure. So to provide nutrients and tissue growth, and also vascularization also, because once you seed cells and prepare like any like functional tissues after implantation, so without vascularization, very difficult to say cell growth. Once you implant, the surrounding blood vessels should penetrate into the porous scaffold. So once it penetrate and form the vascularization. Vascularization means the formation of new blood vessels within this scaffold. Once forming vascularization, these blood vessels provide native tissue environment in order to grow cell and even forming like tissue, functional tissue. That's why vascularization, it is a very big challenge to get like functional tissue. So moreover, it should bond to the host tissue without the formation of scar tissue. Once you implant any scaffold, it should easily bonding with native tissue. It should be easily bonding with native tissue, and it should not form like a scar tissue. It should not form the scar tissue. That's why it should bond to the host tissue without the formation of scar tissue. And also it should resorb at the same rate as the tissue is repaired. So once you implant any scaffold, the scaffold should disappear once it forming like a functional tissue. So that's why it should resorb within the body. So the scaffold should influence the genes in the cells of tissue to enable the efficient cell differentiation, proliferation, and differentiation. The scaffold also should provide the native like environment in order to grow the cell. It means proliferate and even differentiation. The scaffolding materials easily and cheaply produced according to the FDA standards. And also the scaffold should produce a construct with the mechanical properties similar to the host tissue. So if we take an example like a skin tissue, bone tissue. Skin tissue is a soft in nature, bone tissue it is a hard in nature. So based on the native tissue mechanical property, we have to create the scaffold with the similar properties. Like for skin tissue, it requires soft like a scaffold, whereas bone tissue, it requires hard in structure, mechanically strong structure. That's why the scaffold produces a construct with the mechanical properties similar to the host tissue, like bone tissue or skin tissue. So we can classify the scaffolding materials based on the its function, like bioinert, bioresorbable, bioactive. So it should be bioinert. So in some cases, it should be bioinert. It means non-toxic response from the body on implantation usually results in fibrous encapsulation. Bioresorbable undergoes degradation in the body. Dissolution products also not harmful and can be secreted naturally. So bioactive, it produces biological response from the body that results in a bond between the material and the host tissue. So once you implant any scaffold, like a bioactive scaffold, it should behave like it produces a biological response from the body. It means it should easily bonding with the native tissue. So in general, scaffolds are porous structures. So the optimal force size for cell or tissue culture, for example, neovascularization, it requires 5 micrometer porous structure for, whereas fibroblast in growth 5 to 15 micrometer, skin regeneration, it requires 20 to 125 micrometer porous structure, chondrocyte in growth 70 to 120 micrometer, bone regeneration, it requires 100 to 400, depends on the type of tissue growth. We need to prepare like optimal pore size of scaffolds. So for osteoconduction, it requires 200 to 350 micrometer, hepatocyte 82 micrometer, adipocyte 100 micrometer, macrophage 60 to 100 micrometer. So we need to create the optimal pore sizes. Depends on the type of application, like for bone tissue engineering or skin tissue engineering or cardiac tissue engineering, any type of tissue. It requires like a specific optimal pore sizes of scaffolds. So based on pore size, we need to select the manufacturing method. So if you see here, there are like different type of manufacturing method in order to prepare the porous scaffolds as well as fiber network scaffold. For porous scaffolds, we have like many choices like Trojan leaching and combined techniques, solid free form fabrication techniques, emulsion templating, and phase separation and gas forming. These methods, each method can able to produce like different optimal pore sizes. You should see here Trojan leaching and combined technique. So it require approximately like 50 to 1000 micrometer. It can able to produce the scaffold with 50 to 100 micrometer pore size. Solid free form also like approximately 100 to 600. Like depends on the type of manufacturing method. We can able to produce like a different type of optimal pore sizes of scaffolds. So moreover, we can also able to produce fiber networks. For example, electrospun fiber meshes. So it can able to produce at 0.2 to 100 micrometer for structures. So whereas fiber bonding, it can able to produce like 52 to around 600 micrometer for structures. So pore sizes typically opened by different techniques used for the fabrication of porous solids. So what is the porosity? So we are always speaking about porous structure of scaffolds. So what is the porosity? So in general, a pore can be defined as a wide space within the scaffold. You can see here, this is the gray color. It looks, it is a scaffold, and blue color represent the pore. Blue color represent the pore. So it means a pore can be defined as a wide space within this scaffold. Whereas porosity, it can be considered as a collection of all pores within this scaffold. So the pore size and porosity, these two terms are important in order to provide the cell growth or tissue growth. So for example, macro pores like greater than 50 micrometer are of scale to influence the tissue function. Pores greater than 300 micrometer in size are typically suggested for bone in growth in relation to vascularization of the constructor. It means the formation of blood vessels. Micropores, that is less than 50 micrometer, or of scale to influence the cell function like a cell attachment, especially for mammalian cells. Typically, these cells are typically 10 to 20 micrometer in size. That's why less than 50 micrometer enough for the influence of cell function. If a scaffold having less than 50 micrometer, it is enough for the cell growth, especially mammalian cells. These cells have typically 10 to 20 micrometer in size. So nanopores, pore architecture, surface textures on nanoscale, that is 1 to 1000 nanometer. High porosity. High porosity also important. For example, 90% porosity. It provides the greater pore volume for cell infiltration and the formation of extracellular matrix. So interconnectivity, it is also important. Interconnectivity. So 100% porosity and 100% is interconnectivity. It is a choice for tissue, more choice for tissue engineering, because scaffolds for tissue engineering have 100% interconnectivity pore volume, thereby maximizing the diffusion and exchange of nutrients throughout the entire scaffold pore volume. So you can see here how the cell distribution present within this scaffold, like either non-porous hydrogels or superporous hydrogels. So as you know about hydrogel, the previous presenters have explained about hydrogels. So I hope everyone know about hydrogel. So here you can see here these two images, non-porous hydrogels. Here you can see superporous hydrogels. Two images are superporous hydrogels. The cell distribution and composition inside of the scaffold cannot be controlled. However, microstructure of the scaffold should be controlled, because if we seed cells on the non-porous hydrogel surface, the cell growth and expand on the surface of the scaffold only. So whereas if we encapsulate cells within the scaffold, if we encapsulate with a polymer solution and preparing the hydrogel, so it represents the cell encapsulated within the hydrogel. So if you see here, the cell seeding on the porous network, superporous hydrogel, cell growth within the pore only, not on the surface of the hydrogel. So whereas when cell encapsulated with polymer solution and preparing the porous network structure, like superporous network structure, the cells are distributed within the network, not in the pore. So that's why microstructure only should be controlled, not like a cell distribution, cell composition inside of the cell scaffold cannot be controlled. So how about mechanical properties of scaffold? So as I have explained about the mechanical properties of any scaffold should match with the native tissues. In general, our native tissues like cancellous bone, cortical bone, cartilage, ligament, tendon, each tissue having its mechanical property like compressive strength, Young's modulus, and fracture toughness. If you see here, cancellous bone, it's having compressive strength like 4 to 12 megapascal, Young's modulus 0.0 to 0.5 gigapascal. So fracture toughness is not showing. So cortical bone, it having height tensile strength 60 to 160, compressive strength 130 to 180 megapascal. Young's modulus 3 to 330. You can see here various type of tissues. Each tissue having its own like mechanical strength. Bone, it is a very hard in structure, right? You can see the comparison of cortical bone and cancellous bone. Cancellous bone, it is present in the middle part of the bone, right? So it has a less compressive strength because it is like not mechanically strong. Whereas cortical bone, it is mechanically strong. It has like 130 to 180 megapascal compressive strength. So depends on the type of tissue. It has its own like tensile strength, compressive strength, and Young's modulus. So we have to choose the scaffold according to the native tissue mechanical property. So the scaffold mechanical property should match with the native tissue mechanical property. Then only we can apply to the particular tissue engineering. For example, skin tissue engineering. So we need to prepare like soft hydrogels, whereas bone tissue engineering, we need to prepare hard hydrogel or scaffold. So depends on the based on the targeted tissue mechanical property. So why would a medical practitioner like a material to degrade in the body? So in many medical applications, biodegradable materials are preferred because they naturally break down in the body over time. So in general, they do not require a second surgery for removal. because traditional implants often require surgical removal, increasing the patient risk and medical costs. Biodegradable materials eliminate the need by dissolving safely after fulfilling their function. Moreover, the permanent implants can interfere with the natural bone regeneration by taking on too much load, weakening surrounding tissues. Degradable materials allow the body to heal while gradually transforming mechanical stress back to the natural tissue. So, moreover, it can offer tremendous potential as the basis for controlled drug delivery because these materials serve as an excellent platform for sustained and local drug release, helping to deliver medication directly to the target site over time, reducing side effects and improving treatment outcomes. So, from temporary scaffolds to drug-loaded implants, biodegradable materials are revolutionizing regenerative medicine and personalized healthcare, making treatment safer and more effective. So, we need to prepare like for a scaffold. There are many type of scaffolding fabrication techniques that we have. So those are like conventional techniques are solvent casting and particulate leaching and gas forming and freeze drying electrospinning techniques. So if you see here, this is the this is the solvent casting and particular leaching method. So in tissue engineering, fabricating scaffolds with a controlled porosity is crucial for supporting cell growth and nutrient transport. One of the most common techniques used for this purpose is solvent casting and particulate leaching technique. This method is a simple and cost effective and allows us to create highly porous structure that mimic the native like extracellular matrix. So it also provides the flexibility in controlled porosity, pore size, and the distribution by adjusting the progen particles. Because here we are mixing the polymer solution with the progens like sodium chloride salts. So once we mix and then we need to pour on in particular mold. Once we evaporate the solvent, so we evaporate solvent and dissolve and keeping in water. The salt particles are leached out. Particles are leached out. So that the porous structures were created within this scaffold. That's why it can provide the flexibility in controlled pore size and distribution by adjusting the progen particles. Even we can add a different type of progens also, other progen particles also. So the pore size and porosity depends on the size of the progen particles like sodium chloride. So if a progen particle size is higher, then we can able to produce like high porosity and high pore size and porosity. So it is a simple technique. So this technique has some advantage and disadvantage because it requires like it is a solvent casting or particulate leaching technique. So this technique have advantages like it can able to produce the porous 3D form structure and a relative controlled porosity, pore size and porosity. A wide range of polymers can be used because as we know that we have many type of polymers like natural polymers and synthetic polymers. Depends on the type of application. We need to choose the polymers, either natural or synthetic polymers. Sometimes we can combine together also. That's why for this technique, we can use a wide range of polymers in order to create the porous structures. However, this technique has limited disadvantages like it can able to produce membrane type scaffold. The thickness is 3 mm thickness. However, irregular shaped pores because salt progens are not regularly shaped. That's why it can able to produce irregular shaped pores and also poorly interconnected network structure. And sometimes once we evaporate solvent, there is a still present in the residue of solvent that can affect the cell growth, cell proliferation, and differentiation. So this technique has some advantage and disadvantage. So other important technique is fiber bonding technique. In this technique, first a non-bonded fiber structure of polymer simply immersed with another polymer solution, and then after evaporating solvent, then we need to apply uh, heat treatment above the melting point of this non-bonded polymer structure, non-bonded polymer structures. So once we treat heat treat, we do heat treatment, then after that we need to dissolution of continuous polymer B. Then finally we can able to create the bonded fiber structure of polymer A, bonded structure of polymer A. So however, this technique also have some advantages and disadvantages. Like advantages are highly porous and interconnected fiber structure, large surface area, because this type of technique can able to produce like fiber structures. That's why it has large surface area, and also we can improve the mechanical property because the one polymer is bonding onto the fiber structure. That's why we can improve the mechanical property. However, it has some disadvantages like insufficient mechanical strength for load bearing tissues, especially bone tissues. So however, solvent residue, it is always problem sometimes for a cell in order to retard the cell growth and cell growth and differentiation. So freeze drying, it is common technique. So it is known as lyophilization, is widely used for fabricating scaffolds, highly porous scaffold with interconnected networks. In this technique, a polymer solution is frozen. And the solvent is removed through sublimation by using freeze dryer under minus 50, 80 degrees centigrade. And the solvent is removed through sublimation, leaving behind a porous structure. This method is particularly effective for soft tissue engineering, as it allows for the creation of lightweight sponge-like structure, scaffold that supports cell infiltration and nutrient diffusion. So as you can see here, the after freeze dry, the scaffolds looks like highly porous structures. So how about the cell growth within this like a porous scaffold, 3D scaffold. So in cell culture medium, if you immerse within the cell culture medium, the scaffold, which is an open cell foam structure, oxygen and nutrients are supplied from the liquid cell culture. So when you seed on cells on the top of this scaffold, so initially the cell, it can grow and spread and migrate into the pores of the scaffold. Thus, finally, the cells fully colonize and pores and start to lay down their own extracellular matrix. So once the cells on the surface absorb all the nutrients, and the deep part of the movement is stopped due to lack of supply, because the nutrients transportation and oxygen from top of the scaffold to the down part. So this is the diffusion constants of tissue in the scaffold, which are like foam-like structures, foam-like scaffolds. So another important technique is it is advanced technique is a 3D bioprinting. So what is organ printing? So 3D bioprinting is an advanced scaffold fabrication technique that offers precise control over scaffold architecture. It allows for the layer by layer deposition of bioinks containing cells, growth factors, and biomaterials. This method enables the creation of complex patient-specific scaffolds with a high structural fidelity, making it highly suitable for personalized regenerative medicine and organ printing applications. So currently, no real organ has been successfully created by using this technique, but scientists are currently working on this idea and are making progress. So the first commercial bioprinter company is Organova. They they have done the blood printing of blood vessels and cardiac tissue from chicken cells in 2008. So 3D bioprinting, it requires it requires the bio ink preparation. The bio ink, it can be prepared from polymers and by mixing of the polymers and cells. The viscosity, it is very important in order to extrude the bio ink from the using 3D bioprinter. So from this 3D bioprinting, we can able to print all type of functional tissues with the controlled shape. Controlled shape. So currently, a director at Wake Forest Institute of Regenerative Medicine and a surgeon, and their team created a first functional artificial bladder to be implanted in human. So in 2011, they successfully printed a kidney from human cells within the 7 hours. However, the created kidney not functionally in humans, but they are continuously working on this. So other team like Dr. Jordan Miller and Christopher Chen and Dr. Sangeeta Bhatia, they also created a sugar template that can help the safe development of vascular network for artificial organ. So after network is printed, the cells are inserted and network then grow. The sugar template is dissolved after completion of development. However, this technique has more advantage and also some disadvantages also, because the advantages are artificial organ. We can able to print artificial organ for personalized using patient own cells. There is no DNA rejection. It can eliminate need for immunosuppressant drugs needed after a regular organ transplant. So it can eliminate organ donates. So no waiting period, donor waiting period. However, printers cost, the 3D printer cost is high. It is hundreds of thousands of dollars, possibly more expensive than regular organ transplant. So use of stem cell is always still controversial. However, the cost of using stem cells also problem sometimes. So from using this technique, not yet successfully created a complete organ, like a functional organ. So in our research, we develop biomaterials for tissue engineering applications. This means we create materials that can be used to help the body regenerate damaged tissues or organs. There are three key elements in tissue engineering, right? Uh, cells, scaffolds, and growth factors. So our lab focuses on creating scaffolds like a film, hydrogels, beads, porous bonds, or fibers. These scaffolds are 3D structures. These scaffolds are 3D structures that provide a supporting environment for cells to adhere, grow, and differentiate. They mimic the natural extracellular matrix, which is the scaffolding that surrounds the support cells in our bodies. So we design and manufacture these scaffolds using both synthetic and natural polymers. We carefully analyze their properties to ensure they have right physical and chemical characteristics. So in general, biocompatibility, it is a crucial. After creating these scaffolds, we evaluate their biocompatibility through cell culture studies. This allows us to see how well cells interact with the materials, and ensure they are safe and functional within the body. By combining material science, cell biology, and tissue engineering principles, we aim to develop biomaterials that can effectively promote tissue regeneration. So I have always focusing on like fabricating the scaffolds with the different forest networks, such as, especially I have always applied for bone tissue engineering. So bone tissue engineering. So in general, in bone tissue engineering, we need to fabricate the scaffold with a porous structure, either from naturally derived materials, synthetic materials, or bioceramic. Sometimes we need to combine all together to prepare like to prepare like a highly mechanically stable scaffold to meet the bone tissue bone tissue mechanical property. So in general, we use growth factors like TGF beta, bone morphologic proteins, IGF and HGF, etc. in order to grow the cell, in order to differentiate the stem cells to bone specialized cells. So in general, we always find the defected bone, either it is disease or damage. Once we find this disease or damage, then we choose the specific materials, either natural or synthetic materials or bioceramics. Then we prepare scaffolds. Once we prepare a scaffold, then we seed cells, especially stem cells, mesenchymal stem cells on the scaffold. Once we seed on the scaffold, then the cells grow, and for cells to grow and expand within the scaffold. Once these cells expand within the scaffold, then we apply some growth factors in order to differentiate the cell to the specialized bone cell. So sometimes we use sometimes we use direct implantation after seeding cells. Sometimes we implant after formation of functional tissue. So in bone tissue engineering, vascularization it is very important. Formation of blood vessels, very important. Once we implant the particular scaffold, that's why we always focus on the controlling the porous structures of scaffolds. And sometimes we don't use the growth factors. So growth factor free, we apply growth factor free conditions in order to differentiate the stem cells to the specialized bone cells. So once we implant this type of scaffolds on particular defected or diseased bone, then the scaffold produce the functional group. Meanwhile, the scaffold degrade. So why bone tissue engineering important? First, so as we know, humans all getting old, right? As getting older, we start to feel pain, and these are mainly related to bones. So osteoporosis. We can see here, this is a healthy bone. This is a osteoporosis bone. Osteoporosis bone. So this makes it relatively easy to break, even with a small impact, and is not easy to treat. So in addition, joints that have been used for long times are worn out and cause pain and inflammation. According to a report of European Congress Rheumatology, 30% of people who aged 65 or older have knee osteoarthritis. If this osteoarthritis is not treated, it develops joint destruction. So in 2010, more than 600,000 knee artificial joint replacements were performed in the United States. So body start to lose muscle, and it's hard to move or balance, and that will increase the number of patients visiting orthopedics due to falls. And if lost teeth due to aging, they need implant treatments. So if you look at the graph here, the elderly population in 2015 was 12% of the total. According to the aging population, the WHO expects the elderly population to be 22% of the total by 2050. As the population ages, the market for bone surgery and bone tissue engineering will naturally increase. That's why bone tissue engineering is very important for all humans. So as we know, human bones play many roles, right? It represents the foundation for our bodily locomotion, provide load-bearing capacity to our skeleton, and protection to our internal organs like ribs. It also houses the biological elements required for making blood, trap dangerous metals, maintain the homeostasis of key electrolytes. So look at the picture below. You can see the structure of the bone. You can see the structure of the bone. The entire bone is not filled equally. It is not regular. In general, bones are not regularly structured, right? So the surface is made of cortical bone. The surface is made of cortical bone, but the inside is made of a low density spongy-like structure. Low density spongy-like structure. And some bones are completely empty inside. So in general, bone tissue, it consists of about 70% of hydroxyapatite, 30% of collagen by weight. It means bone, it is a polymer ceramic bio-nanocomposite because it has a polymer like collagen, and ceramic material is hydroxyapatite. It is a nanocomposite because hydroxyapatite, it is in the form of nano within the bone structure. That's why bone, it is a polymer ceramic bio-nanocomposite. Also, human bones vary in shape depending on their function. There are long bones like leg bones, and short bones like finger bones, while there are flat shapes like skulls. Now let's see what happens. Their bones are damaged. So you can see the how once a bone fracture, how it can be remodeling itself. This first picture, it represents the it represents the hematoma formation once the bone fracture. This is accompanied by inflammatory reaction, and a large number of signaling molecules like growth factors are gathered. And second one, fibrocartilaginous callus formation. It means the temporary formation of fibroblast cells. And fibroblasts and chondroblast cells. In here, chondrocytes are transported through newly grown blood vessels. You can see the newly grown blood vessels are penetrated within the callus. Then bony callus of spongy bone is formed, and the fracture begin to stabilize. As tissue begin to mature and the substrate begins to mineralize, cartilage cell proliferation decreases. So looks this bone mineralization picture. Bone composite of hydroxyapatite and ions in the body react to form a new bone. So in bone remodeling process, as we know, in bone, there are two type of cells that can play an important role for the bone formation as well as bone resorbing. Here you can see here pre-osteoclast cells. These are bone resorbing cells, bone eating cells. So whereas you can see here pre-osteoblast cells, these are the bone formation cells. Then finally, these cells are specialized. These cells are differentiated into more specialized osteocyte cells after mineralization. So in general, various materials are used in surgery for bone regeneration. Typically metal and ceramic based materials are widely used, and they are often planted permanently instead of bones. For metal, iron, magnesium, for metal, iron, magnesium based materials are widely used. For ceramic, calcium, calcium sulfate, hydroxyapatite, tricalcium phosphate, bioactive gases, and synthetic hydroxyapatites are widely used. So polymers are widely used as composite materials like chitosan, collagen, gelatin. These are all natural polymers used for the bone surgery, and they are biodegradable also. And other non-biodegradable synthetic polymers also can be used for bone surgery. In some cases, growth factors such as bone morphogenic proteins, BMP, and osteogenic proteins are used to promote the more rapid bone growth and healing. So In cell-based method, we guide and enhance the osteogenic differentiation of bone osteogenic differentiation of stem cells into 3D constructs, so that later it can be engineered successfully into appropriate bone structures, appropriate bone constructs. So I will explain the advantages and disadvantages of each material in detail. Metal, sometimes it is used to support broken bones. It is used to support broken bones by inserting metal, replacing hip joints, or for dental implants. This has the advantage of being lightweight and having good mechanical strength, as similar to the native bone tissue. It also has a little reaction with the living body and osteoconductive, because however it inhibits the bone formation markers and have poor osteointegration with the surrounding bone due to difference in strength from surrounding bones. And metals release sometimes toxic ions by corrosion. So it is not recognized as a biological material and is used as a permanent implant. So here you see here there are like several commercial products for bone defects. So ceramics are already used in bone surgery in many ways, because it is used for filling space instead of natural bones, and for joint restoration. It also applies to bone growth, defect repair, jaw and facial bone replacement and teeth. Among the materials, it has the characteristics most similar to bone tissue and has advantage of good osteoblastic adhesion and diffusion. However, there is a still disadvantages, that the tensile and torsion strength is weaker than natural bone, and it is vulnerable to the fracture. So if you see the So polymers also have, if we use polymers, have some advantage, like it has a like great biocompatible and controlled biodegradation. However, it is hardly controllable consistency, weak in mechanical resistance and immunogenic reactions. So in any tissue engineering, we require like three components like scaffolds, cells, and growth factors. In bone tissue engineering, also we require scaffolds, cell, and growth factors in order to construct the functional tissue. For defected or diseased, damaged sites. So in general, a scaffold provides structural support, safe to construct except a cancer cell. Cells grow only in 2D. In general, cells grow in only 2D. So scaffolds are essential to apply tissues in 3D like structures. It provides place for cell attachment and support until it grows. Cells are living part of tissue. It produces proteins and provides function of cells. It gives tissue reparative properties. Growth factors stimulate cells to grow well with proteins and molecules. So by using these three components, we can able to create functional tissues. So as we know about scaffold, it is a three-dimensional architecture. So it can provide to allow the cell adhere, proliferate, differentiate, and secrete the extracellular matrix. So here you can see the picture below is a scaffold made in the shape of human ear. When cells are cultured here, the cells grow in this shape. This is a picture of a cow cartilage cell seed on this scaffold, shaped like a human ear and then implanted under the skin of a mouse. Then the cells grows naturally within the limited shape and cell. Cells grows naturally within a limited shape and size. As a result of this study, the shape was maintained even after the external stent was removed after 4 weeks. After 12 weeks, the cartilage formation was confirmed. The picture on this right side represents a fluorescence images of cells cultivated in a scaffold made with a certain foresize for bone tissue engineering. We can see the cells growing by attaching and proliferating along the shape of the scaffold and showing high biocompatibility. So in tissue engineering, there are the scaffold should have many properties like biocompatibility, structure, biodegradability, and bone growth. So the scaffold used in bone tissue engineering requires several conditions. First, it must be biocompatible. If it is not biocompatible, cells cannot be cultured and applied the body due to toxicity. The pore-rich structure allows the cells and nutrients to move well to the inside and grow. It should also possible to control the biodegradability, control the degradation of scaffold. If the scaffold breaks down too quickly, it disappears before the cell grows. So it cannot provide support, and if it breaks down too late, it may need reoperation to remove the scaffold after the cell grow up, or the cell may not grow completely. So in bone tissue engineering, three important properties three properties are important, like osteoconductive, osteoinductive, and osteointegrative in nature. When designing biomaterials for bone, the three key properties like osteoconductive, osteoinductive, and osteointegrative. These three terms are important. So what is the meaning of osteoconductive? Osteoconductive means the materials provide a scaffold for new bone growth. They do not actively induce bone formation, but create a structure that supports the cell attachment, migration, and proliferation. For example, hydroxyapatite or beta tricalcium phosphate or calcium phosphate ceramics. These materials are osteoconductive because they do not actively induce bone formation. They can provide the new bone growth. So another important property is osteoinductive property. The materials stimulate stem cells or progenitor cells to differentiate into bone-forming osteoblast cells. They can play an active role in bone healing and regeneration. So the common examples are bone morphogenic proteins, demineralized bone matrix. These two are like osteoinductive materials. So another important property is osteointegrative property. So this refers to the direct structural and functional connection between the material and a living bone, native-like bone, ensuring the strong mechanical stability. So it is critical for implants like titanium, dental, and orthopedic implants, which integrate with the bone without fiber tissue formation. So each of these properties plays a vital role in bone repair, regeneration, and implant stability, helping us develop more effective biomaterials for orthopedic and dental applications. So biomineralization, it is the process also important in bone regeneration. Biomineralization, it is a process of deposition of minerals. Minerals means hydroxyapatite minerals on the bone matrix for the development and the self-remodeling of bone. So it is guided by bone cells and proteins. So inorganic crystal growth is important for treating or remodeling the bone matrix. So if we use SBF solution, simulated body fluid, which is a simulated body fluid with ion concentration nearly equal to those in blood plasma, could reproduce the formation of apatite and bioactive ceramics by in vivo. So as apatite particulates are generated on synthetic materials, new bone may form on this mineralized layer. And implants may be stabilized by increasing the material bone interface strength. So without biominerals, it is very difficult to interact with the host tissue. If any like scaffold, if it has like a biomineralizing capacity, it is easily bonding to the native tissue once we implant. on particular targeted site. So moreover, the formation of the formation of blood vessels within the scaffolds also depends on the type of scaffold that has like a biomineralization capacity. So you see here the damaged bone. So it is a scaffold. It is a bone structure with a defected site. So when we implant this hydrogel type, when we fill the hydrogel type material with a black phosphorus, so it could enhance. Initially, it could it could involve the biomineralization to form the hydroxyapatite structures within the within the surface of the hydrogel structure. So once the hydroxyapatite formation on the surface of the hydrogel structure, then it is easily bonding to the surrounding native tissue, so that the surrounding blood vessels are easily penetrate within the hydrogel structure. Then it start to grow the bone structure to repair the bone defected site. So it means if any scaffold having the biomineralization capacity, it can accelerate the bone regeneration. It can accelerate the bone regeneration, because once it produces the hydroxyapatite, it is easily bonding with the surrounding native tissue, because bone, it is a polymer ceramic bionanocomposite. Polymer ceramic bionanocomposite means ceramic material is hydroxyapatite. So once it can able to produce like a hydroxyapatite formation, then it can easily bonding to the surrounding native tissue, so that the vascularization vascularization start to accelerate the bone tissue regeneration. So how do we perform the biomineralization? So in general, we prepare the simulated body fluid. It is a solution with ion concentration close to that of human blood plasma. So we use different salts, various type of salts like sodium chloride, potassium chloride, phosphates, and magnesium chloride, calcium chloride, and sodium sulfate. So we adjust the pH 7.4. So once we prepare the any scaffold, hydrogel or any polymer scaffold with the porous structure, then we immerse, we soak in SBF solution, simulated body fluid solution, and with simulated body fluid solution at 37 degrees centigrade human body temperature. Then the hydrogel start then any scaffold or hydrogel, it start to biomineralize. It means the hydroxyapatite hydroxyapatite can be formed on the surface or within the scaffold under particular time, because it is very close to the human blood plasma, the SBF solution. So you see here, there are like several type of materials that we have for to enhance the biomineralization. So we can use like silica-based material, silica-based bioactive glass, and other order silica mesoporous silica materials. These materials can accelerate the formation of hydroxyapatite formation. So moreover, we can also use the polymers with some specific functionality, like sulfonic acid groups, some phosphate groups also. These functional groups also can easily interact with the calcium and phosphate ions from the SBF solution, that can accelerate the formation of bone-like apatite formation, so via chelation. So in our study, we have used gargum-based gargum polymer. It is a naturally derived polysaccharide, and combined with some phosphate functionality. So this type of hydrogels can accelerate the formation of bone-like apatite formation within the 14 days period. You can see here the same images of morphology of spherical crystal shape of hydroxyapatite formation. These hydroxyapatite nanoform is amorphous in nature, as similar to the as similar to the bone tissue. So I have completed one project from NRF South Korea titled Ultra Fast Bone-like Uptight Formation Growth Factor Free Post Maturation of Bone Cells using Muscle Inspired 3D Hybrid Hydrogels from NRF South Korea for 3 years from 2019 to 2022. So as we know, tissue engineering concept, it requires the combination of cells, biomaterial growth factors to create the tissue for implantable at a disease or damaged tissue. So far, various types of scaffolds are made with a combination of natural synthetic polymers and ceramics for bone tissue engineering. In bone tissue engineering, growth factors can play an important role. Important role for the maturation of stem cells for specialized cells. So we use dopamine. We use dopamine compound in order to accelerate the hydroxy ultra fast bone-like uptight formation within the scaffold. So we have used like natural polymers like carboxymethyl ketone from fungal mushroom, and we combine with dopamine and then prepare the 3D hybrid hydrogels. We use like mesenchymal stem cells. So how the cells grow and differentiate to produce like functional tissue. So in general, growth factors are necessary, but in our case, without growth factors, the hydrogels can able to promote the maturation of bone cells under growth factor free conditions, because these hydrogels can able to biomineralize within the 12 hours. So in another study, we have developed injectable hydrogels. So this is how injectable hydrogels work for bone tissue engineering. First, stem cells are collected from bones, separated and differentiated and cultured in hydrogels or scaffold. This cultured hydrogel solution injected into damaged bone tissue to produce a bone tissue scaffold that is fits perfectly at fractured bone defects. So first, we have used curcumin. It is a phenolic compound that is especially well known for our people, Indian people. So it is usually used for flavoring agent, and also we can use in curry. So we are all familiar about it. This curcumin reduces pain and inflammation and heals wounds and prevents cell damage caused by cancer cells and kills bacteria. Reduces the toxicity of heavy metal. However, this type of curcumin has poor water solubility. It has a poor water solubility and also low bioavailability. So it is considered the potential benefits of curcumin. However, it has been used in scaffolds, hydrogels, and nanoparticles, and it has been used in various fields of wound dressing and tissue engineering and cancer treatment. So curcumin, it is also good choice for bone regeneration because it helps the new bone formation while preventing the growth of cells that reserve old bone. Typically, when a bone is affected by disease such as cancer, various enhancer of gesta homolog to 𝐀𝐓𝐀𝐋 inhibitors are produced, which interfere the bone formation. We found that expression of 𝐀𝐲H2 gene was down-regulated in the presence of curcumin during the bone formation. So to put it simply, it is not easy for a new bone to form in diseased bones such as cancer. This is due to the 𝐀𝐲H2 gene, which prevents the formation of new bone. Because curcumin inhibits the expression of 𝐀𝐲H2, curcumin helps in bone formation. However, as mentioned earlier, curcumin has poor water solubility and bioavailability to treat various bone diseases. So we prepared a curcumin conjugate, oxidized sodium alginate curcumin conjugate, in order to easily solubilize in the water. So then we prepared the injectable hydrogel by combining with gelatin methacrylate by using visible light irradiation condition. So from this study, we observed that greater than 10 micromoles of curcumin, in general, free curcumin it is greater than 10 micrometer. It shows a cytotoxic and pre-osteoblast cells. So in our study, the conjugates with 10 less than 10 micro in our study, the conjugate shows no cytotoxicity and improves the osteoblast differentiation and forms the bone-like nodules. So in this project, we are focusing on developing a novel scaffold for articulate cartilage regeneration. This scaffold will be both eco-friendly and possess the necessary properties of cartilage repair. We use sustainable materials from livestock industry, specifically the mineralized tissues from pig teeth. These bioceramics offer a sustainable and ethical alternative for traditional materials. So we are also working on culture meat applications. So we have team like three different parts of team. One team from Professor Choi from Yeungnam University. They were preparing the media for muscle cell differentiation. So another group from 𝐀𝐓𝐀𝐾 University, they isolate the cells from muscles. So our team, we are able to produce like eatable scaffolds. So we are all team combined and then producing like producing culture meat. So this project is currently ongoing. So I am always focusing on the preparing the alginate based aligned patterns, because the muscle cells grow one direction. That's why we always focusing on the aligned pattern to prepare the culture meat like structures. So we have also developed a dermal pillar in order to cure the wrinkles of face. We have collaborated with Adipogel from Professor Adipogel company. So this is soon going to commercialize this product. So finally, I would like to acknowledge to the Pusan National University and University of Akron and Medcon company, Adipogel company, and some other universities of our collaborators, and from national NRF projects from NRF and a research center for natural products and medical materials. Thank you.
Thank you so much, sir. This time for questionnaire.
Okay.
Sir, one participant asking doubt regarding this topic. Sir, can you explain the effectiveness of the freeze gelation method for the preparation of scaffold?
Can you explain the effectiveness of freeze gelation method of the preparation? So usually we use the polymer solution for freezing. Then it can able to produce like solid structures. Just within the 30 minutes, 1 hour enough for freezing. And then we use a freeze dryer in order to remove the ice crystal forms within this scaffold.
Sir, there is another question, sir. Frozen embryo it's good for fresh embryo. It is good which embryo shows highest rate inflammation, which baby more healthy baby.
So I don't know much about biology because I am chemist. So I'm always focusing on the scaffold fabrication techniques, different type of scaffold preparation, and I know a little about biology. I'm sorry about this.
Sir, now vote of thanks for this section. On behalf of our institution, I take privilege to extend our heartfelt thanks to Dr. K. Madhusudana Rao for a remarkable section by enlightening us the advances in fabrication techniques using biocompatible polymers and their applications. The session was very useful and informative. Thanks, sir.
Okay, thank you. Thank you so much for your invitation.
Have a nice day.
Okay, thank you, sir.
Next section will start with 5 minutes.
Good evening to Chauhan, sir. Please join.
Yes,
Please join the session, sir.
yes, I'm there already. I'm just sharing my slides, so can you just tell me if I'm able to do so or not?
Okay, thank you, sir.
You can see my slides?
Yes, sir. It's visible.
Okay, good.
Sir, please can move the slides, sir.
Yeah, let me just do that. Okay.
Yes,
Let
it's
me
moving, sir.
Okay.
कौन भेज दिया है कल पूरा भेजा हूँ मैं उनको तो भेजा था
Yes. Thank you, sir. Please remove your slides, sir. Can I take introduction of you, sir?
So I think I am able to move slides.
Yes, sir.
So, okay. You're okay with that?
Can I take introduction, sir? A very good evening to all. A warm welcome to Atal Faculty Development Program organized by GATE Institute of Technology, Autonomous Guti. It gives me a great pleasure to introduce today's honorable keynote speaker, Professor Subhash C. Chauhan, sir, who is tenured professor of the Immunology and Microbiology Department of School of Medicine, University of Texas, Rio Grande Valley, Edinburgh, Texas. Dr. Chauhan has successfully established a brand new Department of Immunology and Microbiology and South Texas Center of Excellence in Cancer Research. He has received the STARS Award from the University of Texas. He system and sustainable measure funding from CPRI and NIH RCMI award in addition to several private foundation grants. Dr. Chauhan is also funding director of the South Texas Center of Excellence in Cancer Research. The primary research interest of Dr. Chauhan's lab is to identify and characterize the diagnostic and characterization targets of cancer. The research is aimed for the identification and characterization of biomarkers that localizes in cancer cells to develop newer tools for early disease diagnosis. His previous work has resulted in the publication of high-impacted research articles and significant external funding. Dr. Chauhan has published over 205 papers, reviewed manuscripts and book chapters along with several patents related to drug delivery system, nanoparticle formulations and antibodies. Dr. Chauhan has delivered 80 invited lectures at several national and international forums. Dr. Chauhan has been actively involved in the paper review process of manuscripts for numerous journals, study sections, multiple external funding agencies, training of junior faculties and graduate students. We are very fortunate to have sir with us today and request to you take over the session, sir.
Yeah, thank you very much. Can you hear me? Am I audible?
Sir.
Am I audible?
Yes, sir. Audible, sir.
Good, good, good. Thank you very much. So thanks a lot for this introduction. Frankly, a lot of things even I don't know. I forgot about what you mentioned to me. Thank you very much for recognizing
Welcome,
those.
sir. Welcome, sir.
And I'd like to just thank organizers for giving me this opportunity to share some of my work, what I have done over the last 15, 20 years. So in my lecture, I am going to do two things. One, I'm just going to give a brief background, like what I have done and how, especially for youngsters, I just like to give a story, like how to start a project and how you can take it to a translational and clinical level. So that is going to be one of my story. And as a second part, what I'm going to do, I'm going to give some my thoughts on faculty development and how we can develop a next generation of researchers. So these are the two things I'm going to touch base with, if it is okay with all of you. And again, thank you very much for organizers for giving me this opportunity. So first of all, I'm among one of you, as I am from India. I am graduated from Central Drug Research Institute, Lucknow. And I have been very, very fortunate to always get outstanding mentors. So that's why I really care for mentoring. So having a nice mentor makes your life a lot easier. And this is what I always try to do so. But at the same time, you can see when you are in research, then you have to pay a very heavy toll and you have to work really, really very hard. So from CDRI, I moved to Medical College of Ohio for my first postdoctoral fellowship. And again, I was very fortunate to get an outstanding mentor, Dr. Rajesh Naz. I was there for 2 years. And then until now, I was working in the field of reproductive biology. But after coming here at the United States, I realized like, okay, maybe reproductive biology is not a good fit here. So I moved in cancer research. And again, I was very fortunate to find an outstanding mentor, Dr. Surendra Batra, who is a mucin expert. So I was there for about 4 years, and then I started my own independent career at Sanford Health, Sioux Falls, South Dakota, where I was about for 8 years. And there, from there, I moved to University of Tennessee Health Science Center, Memphis, as a full professor with tenure. And I was there for 6 years. We did a lot of good things there. And after that, I was recruited at University of Texas Rio Grande Valley as a chairman of immunology and microbiology. And I was given a task to develop a cancer center. So this is my research journey so far, and that's where I am right now. So, excuse me. So my research lab work in 4 different areas, and one is early cancer diagnosis and finding out new biomarkers for cancer. Since cancer is a very complex disease, that's why we need to just tackle cancer from different angles. And early diagnosis is one of them. And the second part is cancer prevention. So cancer prevention, like once cancer is there, then obviously we have very limited options to treat the cancer. So better approach is to just develop some preventive strategies so that we are not getting into this mess. So we are working very heavily on cancer prevention area. So cancer prevention include like promoting early diagnosis, having some change in lifestyle modification, like maybe diet, physical exercise, alcoholism, smoking, excuse me, smoking, and ultimately like having a lot of stress. We don't need to put a lot of stress in the body. And the third thing we are looking for, like how we can advance overall molecular imaging of tumors or cancer, and can we develop better way of delivering drugs? Because the way we treat right now cancer, it's very awful, and we don't treat the tumor or disease, rather we do treat the whole body. So the technology what we are developing is more related to like just treating the disease, not the whole body. And fourth area is molecular drivers of cancer health disparity. Again, this is a upgrowing area because different part of the world, they are very differentially touched by different cancers. So in certain area, certain cancers are more prevalent. In certain population, ethnic population, certain cancers are more prevalent than others. So region we don't know, and that's where cancer health disparity comes into the picture. So our laboratory is looking very carefully, especially like we need to cater the geographical need. Where I am right now, Here we cater the Hispanic population, and Hispanic population, due to some reason, we have like a lot more burden of liver cancer and cervical cancer. So this is one of the focus area we are taking in our cancer center right now. So this is just a brief I am going to give regarding my lecture related to science. So I'm going to focus more on pancreatic cancer because this is one of the more mature story of what I have. I don't want to go on different cancers and confuse you, but I will just try to focus on pancreatic cancer. And I'll try to explain you role of a protein which my lab has identified. It's called MUC13 mucin, and how it is involved in pancreatic cancer diagnosis and how we can utilize it for tumor-targeted delivery of drugs and cargoes. I'll also discuss some major roadblocks, what are in pancreatic cancer, and why pancreatic cancer therapy is slim. And then finally, I will just discuss with our laboratory has developed really novel tools that target MUC13, and then we can deliver drugs through our MUC13 antibodies right into the pancreatic tumor. So cancer, I will not go too much in detail, but if you look more carefully or in a precise way, cancer is an abnormal cell growth. It can happen in any part of the body. It's a multi-step process, and it consists maybe more than 13, 14 hallmarks right now. But for ease of sake, if we focus on initiation, promotion, and progression. So these are three main steps in cancer. It need to get initiated, then further need to get promotion, and now next is how to progress. Well, progression means when it is start breaking the nest and moving in other part of the body. So it is a second leading cause of death in human, and they are spending millions of dollars on cancer therapy, cancer diagnosis, but still we are not there where we should be. Although we are making significant progress, but still there is a lot we need to do. Why I'm focusing on pancreatic cancer, or our lab focus on pancreatic cancer? As you can see here, this is in terms of diagnosis, in terms of number of diagnoses, it is on number 10. But I like you to look for the number of diagnosis versus number of death in the lower picture. As you can see here, number of diagnosis is about 18,000 and number of death is about 17,000. So that's why pancreatic cancer we consider a death sentence. And it's very scary cancer, and a lot of time it's after diagnosis, average age is about 6 to 8 months. So that's why still it is being considered as a death sentence. But if we look for the future, in future, even this statistics is more scary, because every other cancer is going to be on declining phase, because we are doing much better in terms of diagnosis and in terms of their treatment. But in terms of pancreatic cancer, it is on rise. And by 2030, it is expected that it will be second leading cause of cancer-related deaths. So you can see why we need to focus on pancreatic cancer. So these are some very famous people who died because of pancreatic cancer, because this cancer doesn't care whether you are a very rich person or a poor person. It is going to treat you equally and not going to show any mercy. So as you can see here, even Steve Jobs, who is a founder of Apple, he was targeted by pancreatic cancer. So what pancreatic cancer, from where it arises? So it's a very small endocrine organ. It's called pancreas. And pancreas, as you can see, it's a very tiny organ in the body, but very responsible organ. As you know, it is responsible for major digestion and responsible for providing major digestive enzymes in the duodenum. So a very small leaflet-like structure. When we even open our mice or human, you barely will visualize it until you're a very expert person. But if it is getting some cancer or carcinoma, then it becomes really, really devastating. And usually it starts from mid or tail of the pancreas. So the problem is the location of pancreas is so terrible, we can even localize it with the help of our physical exam or even MRI or PET scan, right? Or ultrasound, because it is a very cryptic organ. And it's pancreatic cancer is originating and initiating, its overall course is very cryptic, like very hidden. So that's why majority of time, with the help of our existing technologies, we miss pancreatic cancer often. There are certain risk factors for pancreatic cancer, including smoking, diabetes, pancreatitis, obesity, and now we added one more, is stress. Okay. So this is just a molecular pathogenesis, how it happens. So normal duct, they start getting transformed into either pancreatic intraepithelial neoplasias, which we call PANN, or IPMNs or MCNs. And ultimately, their morphology gets changed, that their epithelial cells, they get more columnar, more versatile, and then as a result, it transforms into pancreatic ductal adenocarcinoma. At molecular level, there are certain molecular factors that influence pancreatic cancer. For example, KRAS mutation. In majority of the case, we see mutation in KRAS. We see mutation in P16, mutation in P53, SMED4. In addition to that, we also see overexpression of a lot of molecular factors or oncogene. For example, a fibroblast growth factor, TGF-beta, TGF-alpha, her EGFRs. In addition to that, we see overexpression of a lot of mucins, and that's where my story is now coming into the picture. So we have shown, people have shown that certain mucin like MUC1 and MUC4, they are upregulated in pancreatic cancer.
భలే చెప్పాలి తెలుసా నువ్వు మనోజ్ అయితే ఇంకా బాగా చెప్పాడంట.
Can you repeat your question, please? Okay, so let's see what mucins are. So mucins are, they are very high molecular weight glycoproteins. They are rich in serine, threonine, and proline, and they contain a lot of O-linked glycan chains and some M-linked glycan chains. They are considered to produce by secretory cells, secretory epithelial cells, and in general, like if you go 20 years back, their main function was considered as a protection and lubrication of epithelial cell surface. But later on, we realize now, no, they do a lot more in cancer cells. They are responsible for cell renewal, differentiation, cell signaling, and cell addition. And mucins are a very interesting molecule because majority of the diagnostic marker, if you see, cancer diagnostic markers, they are coming from this family. So that was the reason to focus on this mucin family. And if you look more carefully, mucins can be divided into three categories. One is secreted mucins, soluble mucins, and membrane-bound mucins. And the molecule what I'm going to focus on today is this MUC13 mucin, which is present on 3q21.2 chromosomal location. That's a membrane-bound mucin. Okay, and then so we started this project back in 2005 and 2006. And if you look at a molecular level, this is just a pediatric structure. It has two main domains. One is alpha domain, which is hanging over the cell surface. And it has 10' repeat domain and EGF-like domains, SIA domain. And it also has a beta domain. Okay, so that beta domain is primarily an it is anchored to the membrane, and transmembrane domain it has, and it also has a cytoplasmic tail. And the cytoplasmic tail, it has a lot of phosphorylation motifs. So that's why it can play really a very important role in cell signaling. So when we started this project, there was no monoclonal antibody available on for this molecule. But that time, a group in Japan, they developed a monoclonal antibody back in 2005. And then I requested them if they can share some antibody with me. So they shared a very small fraction, maybe about 100 microgram or so. So from that, I could just generate some of this preliminary data. That's why I always use in my presentation, because this is the initial data what we generated. And it is showing me that, like, really, MUC13 is not present in normal ductal epithelia, but when it starts progressing to panin, or these are early lesions of pancreatic cancer, then it starts expressing, and in a higher stage, its expression is increasing by stage. So I find this data very interesting. That's why we started working on this project. Then later on, what we did, we developed our own monoclonal antibody. I will share some of the data later on with you. And then we further confirm this data, like, okay, in a bigger cohort, is really this data hold or not? Whether MUC13 present in early lesions of pancreatic cancer or not. And indeed, that data holds, and it was showing really high expression in early lesions of pancreatic cancer. Then, as I mentioned, there are two other lesions. One is IPMN and MCNs. And these are primarily like benign. It could be benign and could be malignant. But by seeing that, or by doing seeing their morphology, we are unable to decide, or a physician cannot decide whether we should resect them or just leave them alone. So what we did, we again looked for MUC13 expression in those lesions. And then what we find out, like, if there is a MUC13 expression high, then it means this is going to be aggressive or invasive cancer. If it is a low expressing, then it is a non-invasive cancer. So again, this is a pretty nice finding here. Based on MUC13 expression from this cyst, we can, or a physician can make a determination whether they need to remove that cyst or remove that lesion, or they can just wait for some time. So that is again a very critical data we find from this antibody. Now I'm coming to the pancreatic ductal adenocarcinoma. So there are 5 or 6 different types of pancreatic cancer, and one is pancreatic ductal adenocarcinoma. When we screen those different types of tumors, we find that MUC13 is expressed only in ductal adenocarcinomas, not in any other type of carcinoma. And then we use a lot of artificial intelligence to prove our point, and then look for the band intensity or PCR data, just to see whether a band intensity or expression of a protein can be correlated with tumor burden, tumor size, or its metastatic potential. So what we realize here, like, yes, if there is a high MUC13 expression, that is associated with bigger tumor or larger tumor burden, and the overall expression pattern of MUC13 can be related with poor survival, especially when MUC13 is present in the nucleus. Then it is really a very poor indicator for survival. So based on that, what we did, we are in the process of developing a biomarker detection kit. So it is going to be immunohistochemistry-based kit, and a PCR-based kit, and a ELISA-based kit. So we are in the process of doing those studies right now. So idea is, like, right now, whatever data we have, it is our own center-based data. So what we want to do, we want to do a multi-centered trial with the help of our diagnostic kit, so that we can see the data is coming from different centers, multi-center, and then we will analyze, and that will determine the overall sensitivity and efficacy of our assay. So now, next question we ask, okay, if there is a high MUC13 expression, it's very good, like it can be used for early diagnosis. But $1 million question is what it is doing in cancer cell, how it is influencing pancreatic cancer. So for that, in a biological lab, we usually use a knockdown and knock-in approach. Means like either you overexpress a gene or you knock down a gene and then look for its functionality. So we did that, and then we find out, long story short, like if we overexpress MUC13, then tumorigenic features like cell proliferation, invasion, migration as potential, it goes high. But when we knock down, then it goes down. And at a molecular level, the overexpression of MUC13 influence number of oncogenes or number of genes that can facilitate the tumorigenic features of pancreatic tumor. For example, HER2 is overexpressed with the when MUC13 is high. Its phosphorylation at 1248 phosphorylation site, that is enhanced. So there we can see a number of genes here which are influenced by MUC13 expression. So it means it is doing really a lot of things or making a change at a molecular level in cancer cell. In vivo, if you see, when after conducting all in vitro assays, we finally did some in vivo assays where we used nude mice and we injected cells, tumor cells, which are MUC13 overexpressing versus knockdown. And then again, we see like it is associated with tumor burden. If there is high MUC13, tumor burden is high and mice survival is low. But when we knock down, the tumor burden is low and mouse survival get better. So now I'm getting into not too deep in molecular biology, but at a molecular level, we need to see if that particular protein is doing everything by itself or it is looking for its partners that are involved for further signaling. So what we did, we find out like, okay, that MUC13 physically interact with HER2 receptor, which is also an oncogene. And when MUC13 is overexpressing, it can physically bind to interact with HER2 receptor and can induce the whole MAP kinase signaling in cancer cell. So that's how it is. This is one of the molecular mechanism. That's how it is influencing the overall tumorigenesis in pancreatic cancer. But next question is like once you have a data, now you keep getting deeper and deeper. So we find out this thing. Okay, if there is a MUC13, then which particular domain of MUC13 is binding with HER2 receptor? So then again, it's a question. So we made a lot of mutant deletion construct, and finally find out like, okay, this particular region of MUC13 binds to EGFR receptor. So these are really hardcore molecular biology assays, and we published this paper in Oncogene. Then one other thing, what happened in pancreatic tumor? Pancreatic tumor are highly metabolically active tumors, and they can consume up to 2,000 calories per day. Even a golf-sized tumor can consume up to 2,000 calories per day, and this is all we consume every day as an adult, right? So that's the reason, like a pancreatic cancer patient, they lose weight very easily, very fast, within like 2-3 months, they lose a lot of weight. So we wanted to know whether MUC13 has anything to do with its tumor metabolism or not. So what we find out, like, yes, indeed, MUC13 can influence pancreatic tumor metabolism as it binds with GLUT1 receptor that is responsible for uptaking the glucose into the cancer cell. And that's how it is making those cancer cells or pancreatic tumor more for consuming the glucose. So again, we published this paper. So now what we know, I could not show you all the data, but what we gather so far, that MUC13 bind to GLUT1 receptor, it physically interact with HER2 receptor. It can influence the expression of KRAS although we don't know the exact molecular mechanism, how it is influencing KRAS expression, but we know that it influences KRAS expression and P53 and other proteins expression. So it means what we can do, it means like it's a good molecular target. If we inhibit pancreatic cancer, then all these signaling pathways we can inhibit, right? So we are in the process of finding out some small molecule inhibitors that can inhibit MUC13 expression. We are finding out some microRNAs that can inhibit MUC13. And so these are our strategies, what we are taking in order to utilize the molecular target. So if you look here, one of the microRNA what we find out that can target MUC13 mucin is microRNA 145 and 132. So when we look for, when we repress or when we re-express these microRNAs, then those tumorigenic features go down and it target MUC13 in cancer cells. So then it means like if we give the microRNA 145 into the pancreatic tumor, then tumor burden should go down. And this is what we are seeing here, that pancreatic tumor burden is going down when we are just injecting microRNA 145 into pancreatic tumors. So there are other early diagnostic marker I just want to touch base with it. It's a 𝐀𝐝7. We again last couple of years we identified this particular molecular marker, and it is very, very specific to pancreatic cancer. It doesn't express in any other type of cancer, so maybe we can combine this marker with MUC13, or we can utilize it by itself. So far what I explained to you, that MUC13 is really a key target for pancreatic cancer diagnosis. It overexpresses, it is involved in tumorigenic, and it influences a lot of molecular pathways, including HER2 and JNK and MAP kinase. And 𝐀𝐝7 is another good molecular marker that we can use for pancreatic cancer diagnosis. Now I'm just going to touch base a little bit with how we are utilizing our nanotechnology to deliver cargoes into the pancreatic tumors. So our approach is this way. Like we want to have a delivery system, which is a very smart delivery system. And in this delivery system, we are going to encapsulate drug of our interest. And then we are going to conjugate those nanoparticles with the help of some antibodies or antibodies against some of the molecular targets that overexpress in pancreatic cancer. So it means we need three things in therapy in this kind of approach. One is a very smart delivery system, a smart drug that not only inhibits pancreatic tumor growth, rather it just tackles the whole pancreatic tumor microenvironment. And third thing is we need to have a system or an antibody or a conjugate or a peptide or a aptamer that can hold these nanoparticles right into the tumor site so that they are not going anywhere else in the body. So this is the approach we are developing in our lab, and I'd like to show you some data, whether with the help of this smart system, we can target pancreatic tumor in a much advanced way or much precise way or not. I think in last lecture, you already heard why nanosystems are important, especially in delivering of drug. The main reason I like to mention here, because through these nanosystems, we can achieve a slow and sustained release of a drug. In a typical way, like if you are just injecting free drug, then it can cause a lot of side effects in your body, especially because it reaches to a maximum tolerated drug concentration very easily. And very soon, within few hours, it can come down to a minimum effective drug concentration. So our nanosystem, what they do, they provide a controlled release or a sustained release for over a longer period. That's why our systems are more efficient, and then they can maintain a drug dose for effective drug concentration for longer time. So as I mentioned, like 3 components we need in my smart system. One is an antibody and nanosystem and a drug of interest. So first, since we know that MUC13 is overexpressed, let's see. First step was let's develop our own monoclonal antibody, and with the help of these 2 outstanding scientists, we developed monoclonal antibodies for MUC13 with the help of our hybridoma technology. So we got more than 50 clones. Out of 50 clones, we selected like 3 or 4 of them, which were showing efficacy or which were showing they're just crossing all those barriers in different assays, like immunohistochemistry and flow cytometry and immunoblotting. So long story short, we find out these 3 antibodies that we can utilize for diagnosis and targeting purposes. We also develop a humanized version, because at some point when we go to the bedside, we need to have those humanized version of the antibody. So this is the kind of, now I'm coming to the smart delivery system. So a smart delivery system we developed through our magnetic nanoparticle system. And this magnetic nanoparticle system is patented. And we evolved this system very, very smartly so that it is crossing all those barriers what a typical magnetic nanoparticle might have, such as like they can aggregate very easily, so you cannot control the particle size. Its drug loading capacity is poor, so we enhanced that capacity, and on top of that, we made it much more a viable system than anything else. So we started with curcumin, and in previous lecture I was hearing, you already heard like why curcumin is important. It's a molecule which we use all the time in immune system. So we try to repurpose it for whether we can utilize it for cancer purpose or not, because it has a lot of anti-cancer activities. But again, challenge is how to deliver it at a tumor site more efficiently so that it can do its job. So in a lot of our manuscript and studies, we characterize this system, magnetic nanoparticle system, and we look for its anti-cancer efficacy, which is showing in all in vitro assays and in vivo assays, and we have established its efficacy in our several assays. So ultimately, what we want to do here, our goal is to develop a smart nanoparticle system that we can load with nanoparticles, with drugs or siRNAs, and then conjugate them with the antibodies so that they can find out the right target or right cells, or in this case, cancer cells, and deliver their cargoes into the cancer cell and kill the cancer cell. So that's the strategy we are taking. That's the hypothesis we had. But practically, whether it is possible or not, or viable or not, for that we have done, like we conjugated our antibody with some radioligand, and then because radioligand, then you can image those antibodies, right? And then conjugated those radiolabeled antibodies with the nanoparticle, and then deliver them into the mice. And then we see whether that whole complex can effectively target the tumor or not. So you can clearly see here, with the help of our technology, we are able to deliver our cargoes or our nanoparticle right into the pancreatic tumor here. As you can see here, these are the tumors. Okay. So it means like what we thought, in practical, we are seeing some evidence of that. Like, okay, yes, it is possible. And when we just see, when you see this kind of data, then what we did, okay, just set these mice and put all those organs out into Petri dish and then image those again. So that way you can see whatever you are seeing here, it is indeed the activity you are seeing in the tumor, and you can see clearly here it is in the tumor only, not in any other part of the organs, body organs. So we were very excited by seeing this data, and based on that, we thought like, okay, our antibody can very precisely deliver the cargoes into the pancreatic tumor. And then based on that, we further use those cold antibodies. Before in earlier experiment, it was hot antibody, means like radiolabeled antibodies, but we cannot use radiolabeled antibody all the time, right? So what we did, we just used cold antibodies, and then these curcumin loaded nanoparticle we gave to the mice in alone or in combination with gemcitabine. And you see here, when we give this magnetic nanoparticle in combination with gemcitabine, then almost there was no tumor burden, or tumor burden was inhibited dramatically. It also inhibited the pancreatic tumor metastasis, as you can see here in this data, like here in control mice, there is a lot of metastasis in the abdomen. But when we use our combination of magnetic nanoparticle and gemcitabine, barely there is any metastasis here. So it means like our magnetic nanoparticle system or targeted antibodies, they can clearly inhibit, they can target the cancer cell, and then they can inhibit their tumorigenic and metastatic potential. Then we wanted to see like what is happening. So that magnetic nanoparticle, they inhibit the pancreatic tumor desmoplasia. That is the main problem. And that's how it is softening the tumor and then delivering the drug into the, or giving access to the gemcitabine, getting to the pancreatic tumor more effectively than in earlier condition. So now there is you might have heard about immunotherapy. We wanted to see whether our magnetic nanoparticle system can give more access to the immunotherapy or not, or which are immune checkpoint inhibitors. So these are very, very popular therapies now. Unfortunately, in pancreatic tumor, this therapy doesn't work. But in our system, when we did that, if we use immunotherapy in combination with our magnetic nanoparticle system, then it becomes responsive. So again, this is a huge finding that again, it is allowing that magnetic nanoparticle system, it is depleting the tumor desmoplasia, and that's how it is allowing these immunotherapy antibodies more access into the pancreatic tumor. So here, there is a lot of tumor immune microenvironment change by our magnetic nanoparticle curcumin system, which I will just not go into too much detail, but again, it is showing you that it inhibits the pancreatic tumor desmoplasia. So this system cannot be only used for curcumin, it can be used for microRNA. So we made a microRNA formulation with the magnetic nanoparticle. And the other therapy what we are using, we are also utilizing some of the exosome that can also be useful for delivery of the cargoes. And advantage of these exosome is like these are natural, okay? They are coming from your own body so that you can avoid all the immune responses, okay? And these exosomes, they are already trained to travel in different part of the body, including the tumor. So that's the reason we took those nanoparticles and we utilize a drug, and we find out very, very good result with that. These antibodies, since we have now, we are also repurposing those for drug antibody conjugates. So drug antibody conjugate, that project is really it's going on very well, and we are working with different anti-cancer molecule that can be conjugated with directly conjugated with our antibodies. We are also developing a CAR-T therapy for these. So for MUC13 mutants. So basically, like we have all the information now, and we are very convinced that number one, MUC13 is really a good molecule. It's a good target that we can utilize for diagnostic and therapeutic purposes. So these are our ultimate goals at clinical level. We want to utilize them for our cancer research, cancer diagnosis, and cancer treatment. So now I just want to switch gear. I have only 10 minutes now left. Why teaching and leadership is important. So that's a different area I just like to emphasize in my lecture, because no matter what we do, I have a certain age, right? And after that, I have to just pass on this knowledge to the next generation. Otherwise, that knowledge will just stuck at one point. So that's why teaching and leadership, these are important criteria. So the mission and goal for to develop a standard of excellence in research to improve human health. That's the ultimate goal. Whether I am doing it, or my students are doing it, or my students are doing it, it's a continuous process. We have to keep working on that, right? And so there are few persons I'm very, very impressed with. One of them is Dr. King, Martin Luther King. So in leadership, we should just not be very, selfish. A genuine leader is not in a search of consensus, but a person who can make consensus. That's what the leadership is. And a man all wrapped by himself is just merely a small package. So if you are just focusing on yourself, then it's just, you're a very small package. Okay, you cannot go big. So that's why, like, you have to just think for others. So ultimately, like one of the quote I like to read here, like, leadership is ultimately about creating a way for people to contribute, okay, for something very, very extraordinary to happen. So for any person or any researcher, we need to have some core values. And these core values for any researcher, like honesty, integrity, trust, fairness, encouragement, empowerment, and empathy. So these are really, really some core values what we need to carry if we want to dream big, okay? And any success there are some key components of success. Number one is you need to have a vision. You need to have a right leadership, inspiration, motivation, teamwork, and obviously you have to have very innovative project, right? So these are some of the things. And again, one more quote I like a lot, and I follow it. It is written by Dr. Mr. Gandhi. The day power of love overrule the love of power, the world will know peace. Okay, so we should not just die for the power. Okay, or we should not just it's like we need to just have power, but the power should not. It's difficult to understand. So we should not just love power for overruling, right? Rather, we should just use power for giving love to others. And here, I just want to show you something here. I like to give an analogy. What leadership means and how you, like, all in this game, you can see all the players, and those all players and when they do something good, you realize them, you appreciate them. But there is a corner here, which is the person sitting in the studio or sitting in the room over there, who is guiding all these players what to do. He is not visible ever in this game, right? So leadership is something like that. So again, I like to show you here with some example. So in college setting, I'm giving you example. So you see here, this is a roof, right? And these three pillars are visible here. So students, staff, and faculty members, right? But there is also a pillar here which is not visible, and that's where the leaders are, okay? So leadership is something which is working from behind the scene, which is never visible, but it's still there, and it is responsible for holding the structure. And philosophy for leadership is like we need to be successful, or if we are unable to give the resources to the person who is sitting in the last in the queue, it means our resource sharing or our resource sharing principles are not factually correct. Okay, there are some fundamental mistakes. So, in my opinion, in the field what we are in field of science and education. So these are some of the activities we need to focus on: teaching, research, administration, facility, and outreach. So I think we have been very successful in developing our students in our group, and we need to focus a lot more on curriculum reform, because world is changing and we cannot just keep teaching what we were teaching 20 years before. So we have to keep looking for a new curriculum and make a shift into that accordingly. So in research, here in the United States at least, we have to rely on federal funding a lot, and this is especially NIH, DoD, industry, foundation, and NSF. So we need to train our student and our faculty members so that they are becoming competitive enough to hit those targets in terms of grant. And definitely, I like to share one thing here, which I practice in my group. Whenever you are winning a grant or you are becoming successful, then share that success story with all your colleagues, rather than just showing just your face only, and then you are just not acknowledging your team member. Like here, in one of the story I see, when I joined Memphis, so I win a grant, a Department of Defense grant, and they published this story where I'm sitting with a microscope here, right? After seeing that, and they did not check with me. So after seeing that, I realized like, oh, this is not right. So moving forward, I just told the marketing people like, okay, no, whenever you are publishing a paper, a story related to my research, then please let me know. And then it's not only me, we like to just share the story with all my colleagues and all my group members. So later on, I start putting this kind of pictures, okay, as you can see here. So idea what I'm trying to say here, just share your success with your lab member and your team members. This is always good, and it will help you in team building. So as you can see here, a lot of my student you can recognize here, like you can recognize this, and I'm going to show you why I'm showing this. So now, career development, like Dr. Yelapu is one of my postdoctoral who joined me in 2008. And now he is an independent faculty member. He is an associate professor now and is moving to professor very soon. Again, this is my postdoctoral fellow, Dr. Seema Khan. She joined me in 2012. And now she is an independent faculty member. So all these people are. So they joined my lab as a postdoctoral fellow, but now they are independent faculty members. Okay, so it means like that faculty success and everything, you have to work really very hard. You have to foster into them. You have to invest into them, and keep your policies like open door policies. Make a very foster interdisciplinary collaboration, and be open-minded for the ideas. So for administration, like you have to just work a lot with your governance, and just try to develop all the policies and procedure that can help your younger generation. Outreach activities are again very important, because typically what is happening right now, like whatever we do in research lab, that is staying in the research lab only. So we should just keep doing some outreach activities so that the knowledge what we are getting into the lab, that knowledge is going out into the community. Because again, ultimately that community is our end user, right? So we have to focus a lot more on that. So this is, you can see here, we do a lot of activities, community related activities in our group. This is Purple Night, which we do for pancreatic cancer awareness. We are involved in fundraising, and we do a lot of activities related to like 5K and so these are the things what we need to focus on. And I think I'm done with my time. So these are some of the acknowledgements which I like to mention. I have really a big group, and I'm very fortunate to have that. And I have very committed people in my group. They are working with me for last several years. Some of them are working with me for last 10 or 15 years. So those people are working, and I'm really very grateful to them, and I like to acknowledge them, for all these successes. In addition, all my collaborators, I'm very thankful to them, because without their help, all the work what I have shown you was not possible. And last but not least, all the financial support what we get from different funding agencies, including NIH, Department of Defense, private foundation. Especially, I like to mention Coastal Foundation. They are supporting our pancreatic cancer research for a long time now. And all the lab members who are involved in our group, as you can see, we have a big team of researchers who are day and night working in our group. So thank you very much. And I think I am right on time, just 1 minute extra. Thank you. Any questions?
Thank you, Professor Chauhan. Now, question 8, you have any questions? Participants, you have any questions?
Any students or faculty member, if you have any questions, you can ask me.
Sir, wait for 5 minutes. They are typing in chat box. Okay, now, time for vote of thanks. We are extended our gratitude to Dr. Subhash C. Chauhan, who presented an innovative session on the role of MUC13 in the pancreatic cancer with the relevant details that have greatly enhanced our understanding. Thanks a lot, sir. It highlights the latest advancements. Thank you, Professor Chauhan.
Thank you very much. My pleasure.
Okay, end of the section.